Hello friends, this is Professor Tushar Ugale, working as assistant professor at Department of Agriculture and Entomology. We will go through this session of role of Pseudomonas fluroscence in agriculture. Pseudomonas fluroscence derived from the Greek word that is pseudos means false and monos means the single unit. Pseudomonas fluroscence were initially classified at the end of the 19th century. It was isolated in 1987 out of the agricultural bulk loam soil. It is a rod-shaped gram-negative bacteria and it is a free-living non-pathogenic saprophytic organism which are very helpful for the plant growth. This is the characteristic feature of the Pseudomonas fluorescence to show the fluorescence pigmentation under the UV light, which is the unique characteristic will be observed on the King's medium. The optimum temperature required for the growth is about 30 to 37 degrees Celsius for the Pseudomonas fluorescence for the effective growth. Pseudomonas fluorescence can grow on plant leaves, on the root surface, even rhizosphere. It contribute the plant growth. The significance of the agriculture of this organism is increased because of their ability to degrade various pollutants and toxicants in the soil, even it having their multiple use in as a biocontrol agent against the various plant pathogens. The gram staining process in the Pseudomonas, it is a method to identify the bacterial cultures. A loopful bacterial culture was transferred in on a clean slide. This slide is air dried and heat fixed. The slide was flooded with ammonium oxalate crystal violet solution for the 1 minute. And then the gram iodine stain is gently flooded on the slide for 1 minute. The decoloration of this slide is done by the using of 95% ethyl alcohol. Slightly apply the alcohol drop by drop for the 5 to 10 seconds. Then we can apply the slide washed with the gentle stream of water and stain with the saffron for 30 seconds. After that we can view this slide under the microscope. This is the microscopic view of the bacterial cell where the gram negative bacteria show the pink color under the microscope, gram positive show the violet. The scientific classification of bacteria: kingdom Bacteria, phylum Protobacteria, and the class is Gramma Protobacteria. Pseudomonas, the Pseudomonas order, family Pseudomonas, genus Pseudomonas, and species Pseudomonas fluorescens. Mass production of the Pseudomonas fluorescens can be undertaken in the laboratory with some specific medium, that is the King's Day medium, nutrient broth media, and that is popularly known as the NB media, can be used for the production of this Pseudomonas fluorescens. The material required for the mass multiplication as a media component, here given the Gainsborough media component, here nutrient broth media component, where the peptone is common in both media, only the quantity is different, and the additional requirements like K2H4, magnesium sulfate, glycerol is included in the medium. When we are going to maintain the culture, we have to add the agar at 15 gram per liter of water. This is the procedure of media preparation where you can weigh all the ingredient on the electronic weighing balance. We have to take the 500 ml, that is half quantity of water, utensil, and make the water so little warm to dissolve the all ingredients easily. After heating the water, dissolve the ingredients very thoroughly. Add the remaining distilled water in the mixture and make the volume, make up the volume as a final, which may be 1 liter or 2 liter or 10 liter. Fill this media into sterile bottle as required. We are going to fill 400 ml per bottle. This is a pictorial presentation of the media preparation in laboratory. Plug the bottle with a cotton plug and cover the cotton plug with the help of the plastic paper and rubber band. Keep the bottles in the autoclave for the autoclaving sterilization under the steam with the pressure at 15 psi pressure and about 121 degrees Celsius temperature. When we attain this temperature and the other requirements, we have to keep this autoclave as such closed for 15 minutes. After 15 minutes, we can remove the bottles and we can transfer for the room conditions for the cooling. After that, we can furtherly go for the inoculation laminated flow. This is the how we are going to fill this bottle with a 400 ml of prepared broth. This is the process of autoclaving. The procedure for inoculation in laminar flow: after cooling of the bottle, inoculation can be done with the pure mother culture with the help of this inoculating needle, or even we can use this micropipette for the inoculation. After the inoculation, bottles are transferred to the growth room for the growth. While inoculation, we have to maintain the ratio near about 1/10 of the culture should be added in the liquid broth to get the appropriate growth of the bacteria within 72 hours. Bottle shaking should be done 3 times in a day to help to utilize all the nutrient ingredients in the media by the bacteria. After 72 hours, the growth will be completed. And further testing, microbial analysis of the microbial count and spore formation. After that, we can claim as a complete growth of this organism. This is the method of inoculation in laboratory. If we have to keep the bottle into the growth room for the further growth. This is the bottle showing the growth phase. In left hand side, this is the growth of the first day of the inoculation, and at right side, this is the third day after 72 hours. The turbidity is observed, which having the good growth of this bacteria. Production methods in liquid formation of the Psudomonas fluroscence. It includes the fermenter method, which is used in the highly commercial level in industries. Carboy method is also one batch type fermentation method with help of the plastic carboy. Flask method as a traditional method of this developing this Pseudomonas fluroscence in laboratory. We have to put the flask on the rotary shaker, where there is no need to shake the flask in frequently. This is our method PGPR currently using, that is bottle fermentation method. This is also very popular method in all the biocontrol laboratories in all universities and colleges among the Maharashtra. This is the fermenter vessels, which is facilitated the scale up production near about 100 liter of production. So this capacity is 120 liter. We can go through the 130, 1/3. This is the industrial fermentation setup, which is for the large scale production or the mass production of the bacterial species. Even one fermenter or one vessel can be utilized for the different organisms with the sterilization sterilization methodology. This is the fermenter method. We have to prepare the selective media. We have to pour in the fermenter and start the sterilization process. Cool down the fermenter and adjust the pH. Again, we have to inoculate the inoculum, maintain the proper temperature, and we have to supply air for the oxygen, maintain the oxygen level. And after the 48 hours, we will get the appropriate quantity and the quality of the organism. This is the batch type fermentation system, which is facilitated with the help of the use of carboy. It is also very popular method in the small scale industries, and when we are going to make a consortium of the different organisms, this method is very suitable for the mass production. But only the critical thing is that we have to supply the sterile air for the aeration in each carboy. It is very laborious method, and it can be very sensitive for the point of infection. This is the rotary shaker on which we can set a flask from 500 ml to 5 liter as per our capacity. We can use the RPM level about 100 RPM or 110 RPM during this production about 72 hours. This is the carboy method, where we can follow the all procedure as described previously, the filling of media and sterilization. Even we can directly put this carboy in the autoclave for the sterilization. Only we have to take care to ensure the cotton plug should be very airtight, and it should not be loose, and we should not be give out any kind of the boiling media in the autoclave. This is our traditional method, flask method. It is quite easy in the research purpose or the some small batches, but for the large scale production it is not suitable because it is very expensive, the cost of the glass. It is the bottle method, saline bottle, empty saline bottle method, which is very available in large quantity in scrap. It can be used as instead of the flask. Even we can fill this 500 ml bottle up to the 400 ml in with media. Even it is very easy to handle on a large scale. For the formulation of the WP formulation, prepare the selective media, adds discuss the previously, and adjust the pH and autoclave this media and sterilize media flask in the autoclave. Inoculate the flask in laminar airflow with the pure culture of Pseudomonas. The flask should be kept on the rotary shaker 100 to 110 rpm for 32 degrees Celsius. After the complete growth, after 72 hours, we can add 400 ml per kg of talc with 10 gram of carboxyl methyl cellulose, that is CMC. This powder formulation can be proceeded for the further quality and use in the field. The mode of action of the Psudomonas fluroscence, it facilitates mode of action with some specific its activity like antibiotic production, Siderophore production, induced systemic resistance, competition with other pathogens, hydrogen cyanide production, plant growth promotion like the antibiotic production which help the plant growth. The Siderophore is the characteristic feature which is produced by the Pseudomonas, that is low molecular weight iron binding protein compounds involved in the process of chelating ferric iron from the environment. Iron is among the bulk minerals present on the surface of the earth. It is unavailable in the soil for the plants, is commonly present in the form of Fe3+, which is highly insoluble in soil. So Siderophore, as the Greek word denotes, it is an iron carrier, are small highly affinity iron chelating compounds that are secreted by the microorganisms such as bacteria, fungi, and serve primarily to transport the iron across the cell membranes. Although a widening range of the Siderophore functions is now being appreciated by all over the workers in this field. Many Siderophore produced by the bacteria and the fungi are strong enough to remove the iron from the host binding proteins. This is the main mode of action where it fights against the plant pathogen fungus. It can be used as a PGPR, that is a plant growth promoting Rhizobacteria. Many of species which is used as a biofertilizer or the from the genus Pseudomonas and Bacillus are used as a plant growth promoting Rhizobacteria, which produces auxins and gibberellins naturally at the rhizosphere. It may be used to enhance the plant health and promote the plant growth without environmental contamination. Pseudomonas species are used as a model route of colonizing the bacteria. It also referred as a yield increasing bacteria, which is very popular characteristic of the Pseudomonas. Why it is popular among all the farmers with the distinguishing features like it increase the yield by activating its root system, and even it also give the positive response to the root elongation, which is also reported by the various scientists in this field. This is the pictorial presentation of the how the Pseudomonas fluroscence and other PGPR bacteria are working at the root zone. While as a direct mechanism in the plant growth with a nitrogen fixation, phytohormone production, ACC deaminase production, water soluble B vitamins, P phosphorus solubilization, potash solubilization, and side by side Siderophore production, which helps the direct mechanism of the PGPR activity. Indirectly, the systemic resistance is also increased by the Pseudomonas in the plant, which prevent the plant from the attack of the plant diseases. Even the Siderophore production and competition with the other nutrients, or competition with the space and nutrients for against the host pathogenic fungus. Antibiotics production, SN production, lactic enzymes and toxins production is also another indirect PGPR mechanism which is observed during the growth of the Pseudomonas in the rhizosphere. It can be used as a biocontrol agent where it having the capacity to grow rapidly in vitro and to be mass produced very short period of time. Rapidly utilizes seed and root exudase, which is a very characteristic feature to secrete more auxins and more gibberellins and phytohormones at the root zone. It produces wide spectrum of bioactive metabolites which help to suppress the another host pathogen, compete aggressively with the other microorganisms like the Fusarium pythium in soil. It can be adaptable to environmental stresses and local conditions of the soil. Even it is an inexpensive method that when we use this method, this product once in soil, they don't need to apply more and more, and we can minimize the frequency of application as compared with the chemical fungicide. It can be used as a nematicide. The plant parasitic nematode can be controlled easily with the help of this Pseudomonas fluorescence. Near about it also increase the systemic resistance against the nematodes like root knot nematode, melodon, and species. Even it can be controlled near about 40% of nematode population in a 7 to 21 days after the application. It produces some compounds like phenolic, chitinase, peroxidase, lipase, protease, gelatinase, which help to minimize the attack of the Pseudomonas fluorescence. It can be used as a biofertilizer also. It having the great role in the nematode nutrient uptake also. It also increases the phosphate concentration of the root and shoot compared to the control plant. Pseudomonas fluorescence stimulates the plant growth through the different mechanism as we are discussing the PGPR activity of synthesis of the antibiotics and the production of the plant growth hormones like gibberellins, which increase the phosphate absorption and the other nutrients uptake in the plant. Major role with this activity in nutrient uptake is just minimizing the soil pH by secretion of some organic acids. Pseudomonas having the activity of phosphate solubilization with the making the solubilize the insoluble phosphate in the soil. It actually there are some acids like gluconic acids, formic acid, tartaric acids secreted by these. These acids are secreted by this Pseudomonas fluorescence, which lowering the pH of the rhizosphere, which leads to uptake of the all nutrients and specifically the phosphorus, which is fixed with the calcium carbonate in the form of tricalcium carbonate, which is very easy to dissolve this tricalcium phosphate to this acid activity, which is secreted by the Pseudomonas fluorescence. It also dissolves the phosphatic minerals and chelate the cationic partners of the phosphorus ions, that is PO43 directly. This is the again diagrammatic presentation where all the organic acids which is working on the rhizosphere are secreted by the organisms in the rhizosphere, which activate this mycorrhiza fungi, and even it induce the resistance in the plant to fight against the pest and diseases. Even it prevent the leaching of the phosphorus, which is a very major role to conserve the nutrient in the rhizosphere. The quality control parameter of the Pseudomonas fluorescence, the carrier should be maybe liquid or powder. It should be it can be formatted in the granules also. Viable cell count in the powder formation of granule, it should be 5 to 10 to 7 cells per gram of powder. Even in carrier like liquid, it should be 1 into 10 to 8. Contamination should not be more than dilution with the dilution of this solution. pH should be 6.5 to 7.5 the powder WP formation, and about liquid it can be varied from 5 to 7.5. Moisture percent should be about 8 to 12% in WP formation always carrier. The packaging should be done in the plastic can. In the case of liquid formation, even SDP can be used. Storage should be done at 25 to 27 degrees Celsius temperature. The liquid formation can be stored for the 1 month without any preservative. If preservative can be added, so we can use this formation up to 6 months to 8 months. The WP formation developed on this talcum powder can be used for the 6 months. This formation of WP formation can be filled in the white color bags. These white color bags is very effectively to increase the shelf life up to 6 months. This is the powder formation developed in the Indian Institute of Horticulture Research, Bangalore. The recommended doses, the recommendation of this Pseudomonas fluorescence as a seed treatment 5 to 10 ml per kg of seed before the sowing. Even we can do the instant seed treatment on the field also. Foliar spray, spraying of 2.5 to 5 ml per liter of water applied can be applied through drip irrigation for irrigation method, and it is 100% water soluble formulation can be developed in the different carrier. It Targeted crops like cucumbers and tomato, brinjals, chilies, solanus, vegetables, potatoes, red flowers, orchards, nurseries. Even sugarcane rice can be given this type of Pseudomonas fluorescens formation. Even the perennial crop like pomegranate and grape can be also um, integrated with the use of the Pseudomonas fluorescens. The targeted disease like Pythium, leaf spot, Rhizobacteria, Fusarium, and Anthropos, which are very major bottlenecks in the productivity of the all agriculture crops, can be controlled effectively with the help of the Pseudomonas fluorescens. The method of application, as discussed earlier, we can give through the seed treatment. Even some powder can be mixed with the uh, tuber crops like potato. We can give the seed treatment for the soil borne to prevent the soil borne diseases or the seed borne diseases. The root treatment can be given before the transplanting to ensure the disease-free seedlings or to avoid the disease attack just after the transplanting. We can make seed this formation with the soil about 1 liter with a 40 kg of neem cake or any well decomposed FIM or any compost which can facilitate as a carrier to spread all over the land. Seedling treatment can be given with by dissolving this solution in sufficient quantity of water, can directly applied to the root system of the seedling. Even powder formation can be used for the application in nursery beds for the seedling treatment. Nursery seed bed preparation, before preparation of the seed bed, we can directly apply through this decomposed compost or FIM, and FIM layer can be applied at the top of the bed. Even while watering to the bed, we can use this formation in mixing with the irrigation water and can be drenched through this seed bed. Drip irrigation application, liquid formations can be given direct to the root zone of this plant. Even in powder formation, it can be mixed with water and can be applied 100 ml to 200 ml per plant in the soil. This is all about the use of the Pseudomonas fluorescens in the agriculture. There are various formations developed in the various companies in this field, which are very easy to apply on large scale. This is all about the Pseudomonas fluorescens and its use in agriculture with a detailed mode of action as how it is can be used as a biofertilizer and also nematicide. This is for your educational purpose. It is not developed for the commercial purpose. Thank you.